Where is this space called? Students that they had set up. It's down to the other end.
Oh, so you know.
Yeah. It's just like a switch.
But how are people getting it? So what I don't understand is how are people from out who don't have building access to do that?
That's a great question.
Because that's the part that makes it, because it's inside it's, you know, the room that we use for like events and poster sessions, it's in there. That's why I'm like, unless they have a time machine where they can class, I don't understand that. Unless those doors are.
Maybe they unlock them, but then they didn't tell anybody.
Yeah, coming through the cold.
So I thought.
We know it.
It's gonna be nice to have a body. I have a chance. I know the chance. This will be the video. Yeah, I know. It's cool. Reads like really. This is. Peter's pretty high. Yeah.
Oh, you make sure you got a whole committee. One name.
Yeah.
He has the same link in it. He's not in the department, right?
Yeah.
You ready?
Yeah. Start all release our.
Oh yeah.
Stands over there. Looks like we're about to start. Thanks, Jimmy.
Hi, thank you for joining us, and thanks to you who are online. I'm Shannon McQueenie. I am Nate Evans' mentor. I'm also a vice chair and professor in the Department of Medical Informatics and Clinical Epidemiology, as well as the chief data officer for the Knight Cancer Institute. We're grateful to have you all here today. This is a dissertation defense, and the reason I bring that up is just a couple of reasons. One, especially for those of you online, because we will be able to hear you in the ceiling, please stay muted until the end. We'll have a question and answer thing for the public and everyone in this room and online at the end, but please hold your questions until then. And then we will very politely but firmly ask everyone to leave because we do have a private session after that for his defense. I just want to tell you a little bit about Nate. It's been an honor to work with him, and also I want to give you a little bit of idea of who you're about to hear from. So Nate did his original studies in biological physics at the University of Washington. He then went on and was working as mechanical design engineer at a company called VisionGate, and he was actually working on lung cancer diagnostics. These are normally the things that you think about after you do a PhD, not before. He also was responsible for a patent that was related to cell delivery, precision cell delivery, and then came and decided to do his PhD. So left industry, came to OHSU and began work with myself and Gordon Mills really focused on thinking about preclinical work in terms of how to really drive and facilitate drug development and drug discovery for cancer. And he's going to be talking today about some of the work that he's done. I'm going to warn you, this is a brief snapshot of what he's done. We've had a lot of discussions on how to what to include and what not, because he's done a tremendous amount of work and he's been incredibly driven, and it's been a real honor to mentor him and work with him. So with that, I'm going to hand it over to Nate.
Okay, thank you, Shannon. Can everyone hear that all right? Yeah, all right, perfect. Yeah, thank you. Like Shannon said, my name is Nate Evans. I will be defending my dissertation today with the title, Mechanistic Deep Learning for Perturbation Biology Application to Precision Oncology Drug Repurposing. And so to begin with some of the overarching motivations of our research, we decided to ask the question computationally, how can we improve cancer drug response prediction? And the premise we adopted in our research was that by incorporating prior knowledge into these machine learning and deep learning algorithms, we could improve the prediction performance as well as aid interpretability and improve the generalization of these methods for downstream tasks, specifically in precision medicine. And so today, there's kind of going to be three research outputs I'll present. The first one will be focused on testing feasibility of this premise. We do that with in synthetic data using graph neural network algorithms, where we incorporate prior knowledge in the form of what's called gene regulatory networks. And we're going to be predicting what's called perturbed expression, so kind of a systems-wide perturbation response. Second, we're going to take a slight detour, and we're going to address some of the data quality issues in high-throughput screening data sets, specifically in one of the data sets that we use in our research. We do that with by proposing this method called data evaluation with gradient similarity. And then last, we'll present a couple methods that actually incorporate prior knowledge into these deep learning methods and apply that and evaluate in experimental data. So first, to kind of answer the question, why does this research matter at all, or why do these questions matter? Perhaps unsurprisingly, there is this unmet need in the treatment and understanding of cancer. And last year, cancer was the second leading cause of death in the US, with around 600,000 deaths per year. And the prevalence of cancer is increasing around the world, particularly in low-income countries. And this is a really complex disease, and there are many reasons why treatment of cancer is challenging, but one that we really focused on is that of drug resistance, where we have many drug therapies that work well for the first 3 to 6 months, but over time, cancers often evolve resistance, and that can lead to relapse and potentially to death. And so to kind of highlight just how significant of a challenge this is, there's a 2017 paper that estimated that around 90% of cancer deaths could actually be attributed to drug resistance in one way or another. So addressing this drug resistance is a huge goal for healthcare research. Now, there's a couple ways, kind of a couple lines of reasoning on how we might address cancer drug resistance. Perhaps a naive approach is just to develop more and many treatments. The idea being that as cancer or as patients evolve resistance, as tumors evolve resistance, we can just switch to alternative secondary or tertiary treatment methods and potentially extend treatment for a long time that way. However, a more rational approach is to actually design therapies that specifically prevent drug resistance mechanisms. And so there's kind of two approaches with this. One is to use drug combinations, where we can target multiple biological processes and try to prevent these resistance mechanisms from arising in the first place. And then the other approach is one called precision oncology, where we tailor treatment of the cancer based on molecular features of the cancer itself. And by doing so, we can more effectively and selectively target these disease. However, with all three of these kind of approaches to drug resistance, there's this limitation, which is a bottleneck between basic research and clinical impact, where And this is kind of highlighted by the fact that the grand majority of drugs that enter FDA trials don't actually make it through, less than 15%. And so this really highlights that if we want to address this unmet need in cancer, we need to try to connect the biomedical research and the clinicians, more translational impact. Now, there's another challenge. Kind of at this point, we might think we could just test all possible drugs against all cancers to see if they're therapeutic. However, this is fundamentally intractable simply due to the size or the number of drugs that could be tested. And to kind of highlight how this scales, this combinatorics problem, why it's so challenging, I like this plot at left, which shows the number of drugs that we could choose from. And in red is the number of 2 drug combinations. And green is the number of 3 drug combinations. And so, for example, if we wanted to test all possible 2 drug combinations of, if we have 700 single agents that we're choosing from, that's 100,000 assays. Now, if we wanted to test all possible 3 drug combinations, that's 100 million, and every 4 drug combination is 100 billion, or 10 billion. And so, clearly, this becomes an incredibly massive search problem, and one that we can't actually do with conventional kind of bench assays. And so the only feasible way to approach this is with what we call in silico screening, the idea that we use some kind of computational model that can very rapidly and efficiently screen these drugs and prioritize effective therapies. So the next question is kind of what does this in silico screening look like? Well, there's a couple ways that you can set this up, but this is a common or a traditional approach to this, where we have some type of computational drug response model, and it takes in features of the drug as well as features of the cancer itself, and tries to predict the response of that cancer to the given drug. And by using these like large public data sets, which characterize cell line and mouse model and patient response to these drugs, we can actually learn how the drug features predict cancer response, or cancer features predict cancer response. And so some common features that we use for this, for drug features, we can use drug targets. That's the proteins that a drug binds to. You can also use compound structure or drug-drug similarity. For cancer features, we often use genomics, or these are like gene mutations or copy number variation. We can also use transcriptomics, which is how much of each gene is expressed, or epigenetics like methylation. And in terms of response in patients, we can use features like tumor reduction volume or patient survival. In cancer cell lines, we often use what's called cell viability. It's the proportion of cells that die once you treat it with a drug. Or you can use perturbed gene expression, which is the assay that we're really going to focus on in our research today. And it's the idea that if you have a population of cells and you perturb it with some drug, you introduce a drug to it, you wait some time, and then you measure the change in expression. And so you're measuring many, many genes at some later time point to understand kind of a systems-wide, how has the drug changed the behavior of the cell. And in our research, what we'll present today, we focus on high throughput cell line screening. It's where we have the majority of data, and it's where we can kind of do many of these assays and answer these questions. So let's imagine now that we've developed an in silico screening model and we've integrated into an effective research methodology. One of the questions is, what are kind of the aspects or the attributes of this methodology that would make it useful to precision oncology research? Well, the first one is we want to be able to take in some feature of a cancer or a tumor and predict the most optimal set of drugs that should treat it. It's kind of straightforward. However, there's also this aspect of when we prioritize drugs, we need to understand why they were prioritized. This is an aspect of model interpretability, and it's often overlooked in many of these machine learning in silico prediction tasks. We also want the prediction logic of these models to be kind of mechanistic or aligned with our prior biological knowledge. We expect this to improve the generalization of our predictions. And you can kind of think of it as we want these predictions to be justifiable. If we want to address some of those issues with the FDA clinical trial bottlenecks, we want our drugs to generalize well to patients. We'd like to minimize toxicity as best we can. This is very hard to measure in cancer cell lines, but there's some simple approaches we can take. And considering how large that drug space, the search problem is, we really have to, this process has to be fast and cheap and efficient because we have to scan hundreds of thousands of drugs really rapidly. So let's shift back now to the original premise that our goal is that we want to improve this drug response prediction task, where we have some model, we take in features of drugs and features of cancer, and we predict the drug response. Now, the novelty of our research is that we want to incorporate prior knowledge into this deep learning task in some way or another. And so the first question is, what kind of prior knowledge is actually going to be useful to cancer drug response? And then the second part of that is, if we do identify the prior knowledge that's going to be useful, how can we encode that? What kind of mathematical format can we encode that in that's useful to these algorithms in one way or another? And then on the other side, we need to understand what algorithm, if we do have this data structure that encodes our prior knowledge, what kind of algorithms can operate on that? Because not all machine learning or deep learning methods can. So starting with that first question, kind of what is the useful prior knowledge, we can think about treatment of a tumor as this collection of cells, where some are normal and some are cancerous, and it's this complex microenvironment. But we often simplify this in our cancer drug response models and think about just the response of a single cancer cell. And of this, we know an incredible amount of knowledge. There's many public databases that have characterized the molecular entities that are involved and the relationships between them, as well as how they work together in specific biological processes. And so in our premise of targeted drug response, we can kind of zoom in on individual processes. And we adopt this premise where the drug binds to usually a protein receptor, changes the functional conformation in some way, and this initiates a protein-protein interaction, kind of cell signaling cascade. And traditionally, this concludes an activation of a transcription factor, which migrates to the nucleus and binds to DNA, and then we encode and then we transcribe genes in some way, and this usually changes the phenotype or the behavior of the cell. And so we would argue that this information is critically important to the drug response task, and that if we can include this, we're likely to improve both the mechanism, or make it more mechanistic prediction logic, and improve the accuracy of our predictions. Now, moving on to the second question, how do we actually encode this prior knowledge? Well, fortunately, there's a data structure called a graph. And these graphs consist of nodes and edges, where nodes, in our case, are going to represent the molecular entities, and edges are the relationships between them. And so we can use many of these public knowledge bases to actually encode this large kind of heterogeneous biological network representing, where we actually represent the relationships between drugs and the molecular entities, as well as the outputs that we're trying to predict, which in this case, we're going to be predicting gene expression from a data set called the Link Cell 1000, so the green nodes. Now, finally, we now know the prior knowledge that we think will be useful. We know how to encode it in a mathematical data structure. Last question is, what kind of algorithm can we use? And the one that we focused on initially is one called graph neural networks. This is commonly used in deep learning tasks that involve graphs. And you can think of this as it operates by a message passing function, where you can pass information from one node to another, where in this case, F theta is parameterized by a neural network. And each node has this, what's called a permutation and varied aggregation scheme. And by iterative layers of these GNN convolutions, we can transmit information long distances through the graph. And it's worked very well to make, to predict based on graphs in the past. And to build a little intuition on how GNNs work, I like this GIF where you can see that we have information that starts at this green node and through sequential layers of message passing that information kind of diffuses through the network. And I like this because it's very analogous to our concept of drug response in these biological networks where we have an input signal, which is the drug in this case, and our expectation is that the perturbation should diffuse through that biological network and be kind of mediated by the different relationships. So at this point, we've kind of presented the premise and how we believe that we can address this and incorporate prior knowledge. Now we want to test feasibility. And the first way we did this was by looking at performance of these GNNs in synthetic data. And so we developed this method. We actually extended a package called GeneNetWeaver. GeneNetWeaver uses real-world gene regulatory networks and a method called ordinary differential equations to produce synthetic time series for genetic perturbations. So the idea here is you have this network comprised of where nodes represent genes and the edges represent the transcriptional regulation between them. And if you change one gene in some way, we can see how all the other genes change in response. Now we added a couple aspects to this. We added the ability to simulate cell lines. We do this by starting with a parent gene regulatory network, and for each cell line, we randomly remove a subset of edges. And so this allows us to kind of have a shared, many shared signaling characteristics while also having each cell line have a unique response. We also added the ability to simulate chemical perturbations. And the idea for this is that for each drug we want to simulate, we randomly sample a set of protein targets or gene targets in this case, as well as binding affinity parameters. And we use well-validated models of pharmacological binding to actually simulate how a drug concentration would impact a gene perturbation in some way. And so this is kind of a tool that we can generate synthetic or fake data that follows a set of assumptions, data for perturbation biology. Next, now that we have this synthetic data, we developed a GNN algorithm that was specifically designed for the task. As inputs, we use unperturbed expression, as well as drug concentration, the time, and the cell context, which is, in our case, which cell line we're using, predicting for. And our goal is to predict the expression for every gene in the network sometime after the introduction of the perturbation. And in this case, the gene regulatory network represents the prior knowledge that we're injecting into the prediction logic. In terms of performance, it worked fairly well. On single agents, we could explain around 90% of the variance. On combination agents, it was a little bit lower, explained around 75% of the variance. But I think more importantly, is this method predicted contextual and fairly rational predictions. And that's what this plot at right is showing, where we're looking at the perturbation response of one gene for a given cell line, where the x-axis is time and the y-axis is our gene expression. And the colored lines represent a single cell line, while the gray lines represent alternative cell lines. And so a key takeaway here is that our method is not predicting the same response for all cell lines. This is a critical task for precision oncology. And what's more is that we're fairly accurate, and it's a rational, smooth response. We're not seeing the values jump around. And so this gave us kind of early evidence that, that this premise had feasibility, that we could probably use GNNs to incorporate prior knowledge into the deep learning prediction logic. And our next step was we wanted to shift now from synthetic, aka fake data, to real world data. However, there's some really critical assumptions that change when we go from synthetic to real world. The first being, in our synthetic data, we're using gene regulatory networks, where every edge represents transcriptional regulation. Now, gene regulatory networks are not necessarily mechanistic or causal. They can be correlative. And in our experimental or our real world modeling, we really wanted these relationships to be causal and mechanistic. And so we're using protein-protein interactions and transcriptional regulation through transcription factor knowledge bases. And one of the consequences of this is that gene regulatory networks are what we call homophilic with respect to gene regulation, meaning that like nodes are connected. Protein-protein interaction networks are heterophilic, where we don't necessarily have like nodes connected. And we're going to come back to this because it has some really serious consequences about how GNNs are going to operate. We also have this challenge with prior knowledge, where in real-world data, we have a lot of missing interactions, especially in understudied pathways. And interactions may be either false or they could be contextual in that they're only active in certain cell lineages. We also have this challenge in the synthetic data we were using, gene regulatory networks where each edge has a label as either an activation or an inhibition. And in protein-protein interaction networks, these relationships become far more complex, where they could represent activation and inhibition. They could also represent some kind of multivariate relationship that actually depends on multiple molecular entities, or they could represent some kind of post-translational behavior signaling. And so one of the challenges here is that while we often know when a relationship between two proteins exists, we don't actually know the functional effect of this. And then there are these data quality issues in the high throughput screening data sets that we have to use. In our synthetic data, it was about as perfect as it could be. We had very low noise, and we had high coverage that we knew the time series expression for almost all genes. In the real world data, we measure around 1,000 gene expression, and in the context that there's probably around 20,000 functional molecular entities, that's fairly sparse annotation. And what's more is we only measure at a single time point. And so clearly there are some major limitations to these high throughput screening data sets, and we want to try to address this as best we can. So we're going to take a slight detour and talk about a method we proposed specifically to address data quality issues in large data sets. And to do this, we developed a method called data valuation with gradient similarity. Data valuation is this class of algorithms that characterize the value or the usefulness of data towards some given purpose. So you can imagine if you have a data set and you apply your data valuation algorithm to it, it's going to assign a numerical value for every observation in the data set, where these values represent, a high value is going to represent that it's useful towards some prediction task, and a low value represents that it's either detrimental or trivial. And so we developed this method DVGS, and we showed that low data values actually correlated with things like corrupted samples or noisy features. And so we could use this as a way to filter kind of low quality data. We also showed that when we filter the low value data, you can often improve machine learning performance across a range of prediction tasks. And that's what this plot at right is showing, where our x-axis is the amount of data that we're filtering, and the y-axis is our machine learning performance, where higher is better. And the red solid line at top is our DVGS when we filter low value data. And so as you can see, we actually improve machine learning performance up to around about 30% of the data is filtered out. So that's pretty cool and kind of counterintuitive that you can remove data and improve performance. Now, next we applied it to the Linkcell 1000 data set. This is the perturbation biology data set that we're really focused on. And we saw some kind of conflicting results. We compared to a previous metric of data quality called average Pearson correlation, which is calculated by correlation between replicates. And what we found was that when we filtered data based on our DVGS data values, it did not improve the machine learning performance of these methods. And what's more, it did not outperform just the previous method of APC. However, it did quantify high value data much better, indicating if you filter the high value data, we see performance degrade really rapidly. And so kind of in summary, while we did show that this DVGS method is quite useful on a range of tasks and competitive compared to previous methods, we did not have the utility on the Link Cell 1000 to really justify further application. So shifting back to our original research question, can we include prior knowledge into deep learning to improve prediction performance? Now we're going to apply our GNN methods to experimental data. So we developed this method we called graph neural networks for cancer drug response, or GNN-CDR. And we use cell line omics as inputs. These are going to be node features, as well as drug concentration. And once again, we're trying to predict the change in RNA expression at 24 hours. Now, to inject our prior knowledge, we use the reactome functional interaction network. And we optimize these parameters on a subset of the Link Cell 1000 data set. In terms of results, we had somewhat conflicting results. We evaluated this on 2 different experiments, where each experiment looked at a different subset of the kind of full biological network. In experiment 1, we focused on a somewhat small network of signaling by receptor tyrosine kinases. And we had fairly promising results where the GNN-CDR model outperformed both traditional neural networks and the GNN-CDR model when it was trained on random prior knowledge, which is really important, right? That if we see an improvement in performance on this GNN model, we expect that that's because of the prior knowledge, not some other mechanism. However, in experiment 2, we had conflicting results where the GNN model did not outperform traditional neural networks, and it did not outperform the GNN-CDR model when trained on random prior knowledge, which really suggests that this method is not leveraging prior information well, and really kind of brought into question whether there was utility of this method for prediction of perturbed expression. So at this point, we kind of took a step back, and we wanted to ask, why are these GNNs not significantly outperforming the baselines as we might expect? And we came up with a couple explanations. First, we might have inaccurate or missing prior knowledge, and this would over-constrain the model and kind of force it to make improper predictions. Or it may be that the way we're encoding our prior knowledge, remember we have all this prior knowledge in the form of a textbook, and we squish it into a data structure we call a graph. That may not be as representative as we really thought it was. And then there's also a couple of methodological limitations, and we really focused on these. We identified these limitations by first considering kind of what are the expected requirements of an effective cell signaling model. The first one being the idea of source awareness, where the functional effect of any perturbation on a gene or a protein really depends on the source, on which protein is activating it from, because you could have activators, right? Or you could have inhibitors. And so unfortunately, this conflicts with one of the common GNN assumptions of edge uniformity, which is the idea that any 2 edges that have the same features are going to be treated equivalently. There's also this challenge where the functional effect of edges can be fairly complex and multivariate. For instance, you may need, for a protein to be activated, you may need an activation signal as well as the absence of an inhibition signal. And because of this, these relationships could be challenging to learn. And the GNNs suffer from a few kind of general limitations. The first one is called over-smoothing, which is the idea that GNN node representations become more and more similar to each other as the network depth increases. The other one is called over-squashing. It's the idea that the topology of the network can create these bottlenecks that prevent effective information flow. But the takeaway from these is the over-smoothing and over-squashing can really limit the ability of these GNNs to learn complex multivariate relationships. And then the last element, and the one that I actually think is probably the most significant, is this idea of heterophilic and homophilic graphs. If you recall back when we were using gene regulatory networks, these GRNs were homophilic in that like nodes are going to be connected. If you perturb one node, you'll have this connected component of perturbed genes. Now, if you think about protein-protein interaction networks, post-translational signaling is not necessarily going to impact the gene expression of those proteins or their respective genes. And so what we're going to end up seeing is that a drug will target a protein, and you'll see this kind of long series of signaling with no gene expression changes, nothing measured, and then you'll have some impact on the transcription factor regulons. And this problem is that GNNs implicitly assume homophily, and it's been shown that they don't do well when applied to heterophilic graphs. And so our suspicion was this was one of the major reasons why GNNs are not going to work well on cell signaling tasks. And so next, we wanted to overcome these limitations, and so we developed a method we called graph-structured neural networks. And these were really inspired by structural equation modeling, which is commonly used in the social sciences to test causality between variables, as well as a method called visible neural networks presented by the Eidecker lab, and of course, graph neural networks we've talked about already. But the basic idea is that given some set of prior knowledge in the form of a graph, for instance, here we have a pathway diagram representing our prior knowledge, we can represent every molecular entity in that biological network as a small neural network in our algorithm. And in practice, I use like 5 to 20 channels with just one layer. It's really a very simple function. But then the inputs and outputs of each of these kind of small neural networks are constrained such that they emulate the prior knowledge topology. And so you can kind of see that, for instance, the RAS protein is represented by this small neural network, and the edges go from RAS to PI3K and from SOS to RAS. And so by forcing the deep learning topology to match our prior knowledge constraints, we kind of argue that this will probably improve the prediction logic to emulate true biology. Now, the next question is, how do we actually implement a method like this? We do this with what's called sparse linear operations. And in panel A, we kind of show this toy graph representing our prior knowledge. And function nodes can either be input or output nodes. So input nodes are placeholders for the respective edge. So I0 and E0 are equivalent, and E5 and O4 are equivalent. And each function node, or the yellow nodes, are encoded or parameterized by a neural network. And if we focus on just function node 1 for a moment, we can see that in this toy graph, we're encoding it as a two-channel, one-layer neural network with inputs from edges 0, 2, and 3, and outputs to edge 1 and 2 at the next layer. Now, to kind of scalably and efficiently implement this, we can update all edges at the same time using this sparse linear operation. And to understand how that works in panel B, the function node 1 that we just discussed is shown in red. And we can see that it has edges from E0, E2, and E3, as well as E1 and E2. And so by stacking these GSNN layers, we can actually propagate information a long distance through the graph and make effective predictions. This overcomes many of the limitations of GNNs that we no longer make the assumption of homophily, and we can learn really complex multivariate relationships between the inputs and outputs of each molecular entity in our graph. Previous to our other methods, similar to our other methods, we encode this data structure representing a biological graph, which includes drugs, proteins, and RNAs, and we're predicting gene expression changes. In terms of performance, we saw fairly promising results where the GSNN outperformed traditional neural networks across all of our experiments. as well as outperformed the GSNN when trained on random prior knowledge. And also, these last three columns are traditional graph neural networks that we use. And they perform quite poorly, kind of supporting our rationale for these GNNs probably not working well on cell signaling tasks. We also wanted to look at what we call local performance, where we group performance by specific attributes. And in this plot at right, we're grouping by different drugs, where every point represents a drug, and the x-axis represents the GSNN performance, y-axis represents the neural network performance. And so any points that lie on the lower triangle are better predicted by the GSNN. And so as we would kind of expect from our previous slide, the majority of drugs are better predicted by our method. However, there are a number of drugs that are better predicted by the neural network. There's this kind of cluster in the lower left where many drugs are neither predicted well by the GSNN nor the neural network. This is probably a data quality issue. However, there are other drugs like this one at the upper left is cobinetinib, where it's better predicted by the neural network. And so one of the questions we have to ask is, why are some drugs well predicted and others are not? And one of our explanations is it could be that the prior knowledge has kind of a selective usefulness to certain drugs that, for instance, if we're missing drug targets, which represents prior knowledge, it's not going to be well encoded in our network. If we're not including kind of the biological processes or the pathways that are used in drug response, we're probably not going to be able to predict it efficiently. Or maybe this aspect of we really assumed this drug target premise where a drug binds to a protein and enacts some kind of change, but there's an alternative approach, which I usually call drug condition premise. It's the idea like DNA damage drugs that create lesions on the DNA, and then a protein sensor detects the lesions and creates some kind of cell signaling. And so for drugs that create functional changes in the cell based on these conditions rather than on protein binding, we're not going to be able to model those well at all. And so this is a really important way, especially for future development, that we want to refine model to understand how do we kind of select effective or useful prior knowledge, and how can we improve the architecture to leverage it properly. Now, we also had this expectation that if we were effectively using prior knowledge to make more mechanistic predictions, downstream prediction tasks should also be more accurate and more generalizable. And so we did this chain prediction task where we predict perturbed expression using our GSNN. So in this case, F expression can be either our GSNN model or a neural network. And then we use a small neural network for F viab to predict viability directly from perturbed expression. So this is cell viability is the proportion of cells that die given a drug. And what we found was that when evaluated on single agents, it didn't really matter if you used the GSNN or the neural network, you got fairly comparable performance. However, when you look at drug combinations, two drug combinations, our GSNN method works markedly better than traditional methods with an MSE less than half that of the neural network. And so this really supports our concept that downstream prediction tasks of our mechanistic model should be more generalizable. We also wanted to provide methods for interpretability of our model. And so to do this, we developed an algorithm we call GSNN explainer. It's actually inspired by a method called GNN explainer. And it's the idea that for any prediction that we make with the GSNN, we can explain the prediction in terms of this compact subgraph of critically involved edges. And that's what this plot at right is showing, where this is explaining the drug response of a proteasome inhibitor in a prostate cancer cell line. And so at the top, you can see the drug. And then we have this series of proteins involved in signal transduction. And then the green and the cyan dots represent RNA predictions. And so really importantly, this graph can be conceptualized as a testable biological hypothesis for this drug response. For instance, we could actually go in and ask, is TP53 actually involved in this proteasome inhibitor response, and test that at the bench? And so this potentially could be useful for basic biological research. It could be useful for building trust in the GSNN predictions, to kind of ask a biologist, is this really what happens? Or we could use this to tune model performance in the future to make sure that our prediction logic actually does align with prior biological expectations. I also wanted to note that traditional methods do have ways that you can explain predictions. However, one of the key factors is that, for instance, neural networks, they do not explicitly model any of the latent molecular entities. And therefore, it's impossible for them to create an explanation in terms of those latent entities. And so this is a really useful and novel approach in explanations that traditional methods cannot do. Next, we wanted to actually apply our GSNN method to drug repurposing. And so to do that, we developed an approach that is kind of built on the premise that if any drug kills all cell lines, it's probably just going to be toxic. It's not going to be therapeutic in any way. And so our idea was that we want to prioritize drugs that create a selective response. And so we do this by specifying a set of target cell lines. These are the cell lines we want to see a response in, and a set of background cell lines, which we want to see no response or less of a response. These are kind of surrogate for normal cells. And by doing so, we can then use our GSNN model to predict the cell viability for each one of these sets, for the target cell lines and for the background cell lines, and prioritize drugs based on those that create the maximum selective response. We evaluate this in using FDA drug indications. And these FDA drug indications are, for every drug which is approved, there's an indication for the disease that it can be used in. And so, for example, if we set the target disease as non-small cell lung cancer, then we take all cell lines derived from non-small cell lung cancer, and we use the background disease of acute myeloid leukemia. And so our expectation is that an effective prioritization should prioritize all drugs with indication for non-small cell lung cancer for drugs with indication for acute myeloid leukemia. We quantify that with the area under the receiver operating curve, where 1 is a perfect prioritization. And what we find is that our method actually works quite well with almost perfect prioritizations across the board, except 2 cases where it's not imperfect or not perfect. And it outperforms the neural network in several tasks. We also went in for the 2 cases where we didn't have perfect prioritization. We did a literature review and actually found that many of the drugs that were kind of quote unquote misprioritized actually had supporting evidence for that they could be repurposed in the alternative diseases. So that's kind of a limitation of this FDA drug indication approach. And then last, we asked the question, will this drug repurposing work for drug combinations? However, unfortunately, FDA drug indications aren't really assigned for, there just aren't as many drug combinations yet, so we can't use the same approach. However, we did perform drug combination prioritizations for breast cancer, and then we did a literature review to look at, for the top prioritized candidates, is there supporting evidence for that? And we found that a majority of them did have supporting evidence suggesting that this method has utility for drug combinations as well. And so kind of in conclusion, we developed this GSNN method that works really quite well at leveraging prior knowledge to improve the performance, hopefully by creating this mechanistic prediction logic. We also showed that we could use this method to aid interpretability, and that it's more useful on a range of precision medicine tasks than just traditional deep learning is. We have some ongoing works in the Mills lab. We've prioritized a set of drug candidates, and we're actually testing those at the bench. So very excited to see where that goes. And in terms of future work, there's some really exciting ways that we can refine this algorithm to continue to improve performance. And one aspect that I'm particularly excited about is combining the GSNN method with something called neural ordinary differential equations to apply it to single cell perturbation biology. So with that, I'd like to acknowledge a lot of people, particularly my advisor, Dr. Shannon McQueen, who's been awesome and supported me these 6 years, as well as my advisory committee and the many DMICE students and faculty and staff, and of course, my parents and friends, who many of whom are here today. And with that, I will take questions. Gareth.
I have a question about the prior knowledge, the sort of randomized prior knowledge.
Mm-hmm.
Do you think if you included patients do you think that the prior knowledge in the randomized network or is it trying to force to emulate sort of what we know?
I don't have any expectation that combining models built on true prior knowledge and random prior knowledge would improve performance in any way. One of the reasons why I suspect that these GSNNs operating on random prior knowledge still have some predictive performance is that in many ways it's flexible enough that it can learn response patterns, even if the randomized GSNNs are unlikely to be mechanistic. They could still learn. They're still kind of non-parametric in certain ways, or enough. The flexibility is there to learn accurate responses. But it's one of the challenges because we want to use that as a baseline for false or random prior knowledge, and yet how do we ensure that these edges don't exist in real life? Cool.
Great work. So in either your data evaluation portion or in your comparison of your GSNN versus traditional neural networks, I was curious if there's any bias towards certain pathways that are better predicted by your network, or pathways that are dropped out in your data evaluation, if you use that knowledge to kind of ensure high throughput screening.
Yeah, so the choice of prior knowledge is I think of it as a really critical hyperparameter to a model like this, where you have to select the prior knowledge that you think will be useful. And so we know that there are huge biases towards certain pathways. And if, for instance, if we try to predict on understudied pathways, I suspect this model will perform very poorly. And so that was really the motivation for why we tested on multiple experiments, where each experiment was actually focusing on different subsets of the biological network. And what we wanted to show was that we kind of had pretty decent performance in these known pathways. I think a much larger question to ask, and one that we'd like to kind of investigate in the future, is how do we characterize performance based on, like, how does prior knowledge correlate to performance, and how can we use that to kind of optimize the selection of the prior knowledge that goes into these models?
If I understand correctly, the model still doesn't take into account whether the edges are inhibitory or active.
Can you online me hear the question? They only hear you.
Totally. Yeah, the question was asked, the GSNN model that we developed, they still don't take into account activation versus inhibition edges. And That is true in the prior knowledge that we don't encode whether an edge is an activation or an inhibition. However, the way we've set up this GSNN model is it learns the functional effect of an edge from the data that it's optimized on, in this case, the Link Cell 1000 data set. And this is actually another really exciting future direction is we haven't done this yet, but it should be possible to go in and look at these individual relationships between different proteins and try to understand how does one protein affect another? Is it an activation edge? Is it an inhibition edge? Is there some kind of more complex multivariate thing going on? And so no, we don't take into account the prior knowledge of activation versus inhibition, but the model itself is assumed to learn that.
You're sticking with such a complex problem, and there's getting to be more and more data out there, so you keep at it and you get closer to making sense of it, I'm sure.
I think this is an extremely important. I think one thing that is helping my knowledge. I have two questions. The first is, so you talked about the heterophilic structures and how the GSMN, that is one of the things that may have really made it such an appropriate model. Are there other areas where you think that type of structure exists, maybe even outside of the domains of biology? And do you think that this model could be useful?
Yeah, so the question was asked, one of the motivations for the GSNN was to apply it to heterophilic networks. And are there alternative domains that you could apply the GSNN to that might also work well? I think that there's a couple simple applications that, for instance, if you wanted to model something like water flow through a pipe or electrical circuits, I think the GSNN would do really well. I don't know if it's really neat for a task like that. However, one domain that I do think this could be applied to effectively is causal modeling. So for instance, structural equation modeling, which kind of inspired much of this work, usually assumes linear relationships between different latent variables. We could use the GSNN to encode known relationships through models that can take into account multivariate relationships. And I think that could be really useful in a lot of tasks. Kind of causal modeling spans almost all domains, and so it could be very widely applicable.
And the other one was mentioned that there still might be, I think, with every model. You see it's kind of permanent. How do you think you might be able to overcome?
So the question was asked, in this model, what are the most striking limitations? And there, I think there's probably many. There's many things that I'd like to address. The aspect that I think could most improve performance, the kind of low-hanging fruit, is the selection of prior knowledge. It's very challenging to know for any given drug what biology is involved in the drug response. So I think that there's ways that we can do this, something called hyperparameter optimization, which would almost require just iteratively retrain the model many, many times on different subsets of biological networks and see which one works best. And so by doing so, we could actually kind of narrow in on what processes are critically involved and useful and which ones are not. And then the other aspect that I'd really like to include, I touched on it briefly, the idea that we assume a drug target premise, that a protein or a drug binds to a protein in an exome change. I believe that there are many drugs that are better encoded with this idea of drug condition, that a drug changes the conditions of the cell and then enacts some kind of functional change. And so I think that that's a shift in how we approach that underlying prior knowledge network, that we need representative nodes that represent conditions rather than just molecular entities. So there's kind of a wider discussion on maybe more representative encodings of the cellular states of the cellular system. Yeah.
So, yeah, that piece of prior knowledge was really. Is there any way to quantify how confident you are in the prior knowledge? And then, in that case where the second experiment, no prior knowledge, or like the randomized network without prior knowledge did well.
Oh, in the GNN?
Yeah.
Yeah, the second experiment, the GNN did best on randomized prior knowledge.
Yeah, does it speak to that piece of prior knowledge? Do you feel like the first model, like, I don't know, was it more solid network or?
Yeah, so the question was asked in our work with GNNs, the first experiment did well and the second experiment did poorly. And the question is that an aspect of the prior knowledge that went into it? I think that it probably does have a huge factor. It was very challenging to understand, is it the prior knowledge going into it, or is it a methodological limitation to kind of like separate those two? We don't have a good way to quantify if performance is being hindered by the prior knowledge or being hindered by the methods. And so it's a challenging one to answer. I don't have a good summary. Yeah.
So with DMICE, one of the things that we stand apart from some of the and you can imagine essentially having a model and using DMICE to essentially do the foundation, then using those values to support classification ahead of retraining the model. So essentially, we either do the GS and the model, we get those values, and that model itself can be used to reparameterize future model.
Yeah.
Essentially in the life cycle. Are there any use cases where we could see that being applied?
Yeah, there's a whole domain on that exact approach. It's called it's usually called like reweighting optimization schemes, I think is the term thrown around. But there's a couple of papers that have shown that works fairly well. I haven't considered using the DVGS method for that, but I suspected it would work quite well. And it could be a way to kind of rapidly combine addressing data quality and creating this effective machine learning model all in one.
Yeah, because it seems like straights.
Yeah.
Especially when you so essentially
Thank you. Yeah.
We do have a question in chat.
Okay, we're right at time, but we can take it. Karen, you want to read it?
Yes, this question came in from Rory. Great presentation, Nate. I'm assuming you can't make any changes to how the drugs actually perform. What's one change you would like to make? Data types, data annotation, data connectedness that you would think would stand a chance of drastically improving your model. Pretend you can ask for anything.
Well, thanks for the question, Rory. If there was one thing I would change with the model, I really want to shift from what we used a data set that's called bulk sequencing. I really want to shift into single cell sequencing. I think that it offers a lot of potential to improve the performance and think about these drug responses in more useful kind of ways. So if I had to choose one, it would be shifting to single cell RNA sequencing rather than this bulk sequencing from the Link Cell 1000.
Great.
Okay, thank you.
Thank you, Nate.
You're getting some comments thanking your presentation. Shannon, at this point, are you asking everyone to step off?
Yeah, I already did. I don't know if you heard my introduction. Karen, we're going upstairs, so we're gonna, yeah. Thank you, Karen.
Thank you. Well done.
Oh.